think about like this pathogen right now being like a robust, like car or truck where you can either use for a vacation with your family or to plow the snow in the winter, or to pull a trailer with pigs or cattle. So it's like a very equipped car that we're noticing right now that it's circulating out there. And obviously, like I said, with other factors that can contribute to more severe disease in post-wean pigs.
Welcome, everyone, to another episode of the Swine Canada podcast show, brought to you by Wisenetics. We are dedicated to bringing you the latest and greatest in the Canadian pork industry. I'm Miranda Smith, and I'll be hosting today's episode. And today's guest is Dr. Rodrigo Paiva, who is a veterinarian and is currently a PhD candidate at Iowa State University. So welcome to the show, Rodrigo.
Thanks, Dr. Miranda, and thanks for having me.
Yeah, so today we're going to be talking about post-weaning colibacillosis, which I'm very excited about because, as I already mentioned to you before, I know nothing about this topic, so I'm ready to learn something today. But before we do that, can you first start by introducing yourself?
Yeah, like I mentioned, my name is Rodrigo Paiva. I am a DVM from Brazil. Was born and raised in a small town in the southeast of Brazil, and that kind of shaped myself to the vet school. And then it's been 13 years now since I graduated, and I had the opportunity to practice down there in Brazil for almost 8 years with the swine industry. And then in 2021, I came to ISU to do master's in PhD. And like you mentioned, my PhD has been focused on colibacillosis, post-weaning colibacillosis. We started to see a couple of years ago an increase in the cases associated with E. coli post-weaning, and over the total cases of diarrhea here at the lab, and that kind of guided us to do some research on that to try to understand, what was going on, what has changed that made those cases to go up.
Your herd software spits out 50 metrics. Guilt asks 3 questions. Which 10 actually matter? Who owns each one? And what happens this week to fix the red ones? Because every human being procrastinates. Good deadlines fix that. Guilt gives your farm a weekly pulse and a quarterly pulse, so the whole crew's energy pulls one direction. 3 pilot farms this quarter, 2 spots left, 1,000 sows or 2 sites. GetGuilt.com.
Okay, so I'm looking forward to be talking about that. But before we do that, can you first kind of start with explaining what is colibacillosis? How do you recognize it? What is it caused by? A little bit of explanation on that.
Sure. I think, colibacillosis can be neonatal in pigs and also post-weaning. And it's caused by a pathogenic E. coli. We have commensal E. coli, which is harmless to piglets, but we have pathogenic ones that they usually harbor different virulence factors that made them pathogenic, including fimbria, adhesins, and toxins or genes that encode for toxins that is going to cause or induce secretory diarrhea, either, like I said, in neonatal piglets or post-weaning. And it's typically a multifactorial disease. We have predisposing factors. We have these virulence factors, like I mentioned, that are required that the pathogenic strains have to be able to occur the disease. But most of the time, when we have cases here at the lab, they are associated with changes in diet or changes in the environment that those pigs are, inevitable stressors that we have at that weaning phase, that all of those kind of contribute to the disease at the end. But the virulence factors per se, they are kind of required for that to happen. So what we've been doing lately pretty much is trying to understand what has changed from the bug side, from the bacteria side, that triggered like more severe, moderate or severe disease associated with that post-weaning phase.
So yeah, like as you mentioned already, like there's a lot of different factors that can cause post-weaning diarrhea. So like in how many of those cases would you say that colibacillosis is a factor?
Yeah, we have viruses that can cause obviously enteric disease in pigs. What we see here, especially in US, especially uh, I would say starting 2016 or 17 till now, we saw an increase in cases associated with E. coli, with pathogenic E. coli, over the total number of other causes of diarrhea in pigs, either viruses or non-infectious causes as well. And that kind of, raised, with that, we start to raise some questions. What has changed? And even including other bacteria as like salmonella, and viruses like coronaviruses. So comparing all of those, we saw that E. coli, pathogenic E. coli, started to stand up in terms of being associated with cases, and that's why probably and that's what made us to kind of go back and try to understand either retrospectively what happened, and also from the pathogen characterization as well, if something has changed over the past years.
So do you have like an actual idea of the prevalence of the colibacillosis? Like, is it like 20% of the cases with post-weaning diarrhea, or like 50 or 5, or like, do you have any idea about that?
Yeah, what we did in one of our studies, we did kind of a rate of the total number of colibacillosis over the total number of diarrhea, and then the rate kind of increased significantly for different years. We saw a overall increase after 2016, but especially in 2021, in 2022, we had a significant increase, and that the rate increased like almost 30% over the total number of cases. So we did see that increased. We do not know by this point exactly why that happened. Like I said, is multifactorial, but at least from the pathogen standpoint, we already know that we had some changes that we are pretty sure that contributed a lot to that increase.
So can you talk about those changes?
Yeah, sure. E. coli, and I don't think that's something that is only happening right now, but I just think right now we have like better tools to better to understand those changes from the pathogen side, and with that we used whole genome sequencing from multiple isolates starting from 2015 to this point, to see if over this time we had changes in terms of virulence factors, and we did. Comparing with Canada, for example, where we have, in my understanding at this point, a lot of F4 phenotype and F18 phenotype circulating in the field. Here at US, F18 has increased and overcome the F4 cases, especially related with post-weaning phase, and it's much more predominant right now, the F18 phenotype. And then within that F18 phenotype of E. coli, we have multiple virulence factors that we noticed that the pathogen started to acquire in the past years and became more virulent. And what I'm trying to say is, think about like this pathogen right now being like robust, like car or truck where you can either use for a vacation with your family or to plow the snow in the winter, or to pull a trailer with pigs or cattle. So it's like a very equipped car that we're noticing right now that it's circulating out there. And obviously, like I said, with other factors that can contribute to more severe disease in post-wean pigs. But so we have the fimbriae, right, which are finger-like structures that the bacteria has to kind of adhere to the enterocytes and then release toxins to cause secretory diarrhea in pigs in that phase. And then together with this fimbriae type, we are observing other toxins like LT, STA, and STB toxins that are associated with the secretory diarrhea, but also other virulence factors that contribute to the bacteria survive longer in the environment, survive longer even within the host, with like serum resistant genes and all that. So the majority of them also being lately hybrid pathotypes. So we are going now in classification of E. coli, but we classify E. coli in pathotypes, different pathotypes like enterotoxigenic E. coli, ETEX, Shiga toxin producing E. coli. And though that classification is pretty much based on this virulence profile that they have and the clinical disease that having this profile, this bacteria is going to be able to induce. And the hybrid ones, which are classified with like containing virulence factors that are associated with enterotoxigenic E. coli and Shiga toxin producing E. coli. So ETEC and STEC ones, they has been more predominant lately, which means, like I said, this car, well-equipped car or truck containing genes, that are making this bacteria being able to cause either secretory diarrhea or even edema disease, the Shiga toxin producing one. So it's a pretty virulent strain, or I would say strains, within this category or hybrid pathotype that we saw that it changed, especially after 2016. And, some of the genes through whole genome sequencing, we were able to kind of understand that were probably acquired from previous, clone or even different clones. So the bacteria is pretty much hanging on there in the intestine of the pigs, right? And they're being able to survive longer in that environment. And that is, let's say, creating more opportunities that where the bacteria can kind of share virulence genes between different strains. And then the pathogen also wants to survive, right? That's your truth. And that is kind of contributing to this pathogen evolvement.
So how do pigs get in contact with these bacteria? Like, are they, because like, let's say they're being moved into a nursery that it's squeaky clean. So how does this bacteria somehow make their way into these pigs?
Yeah, that's a very good question. Going back to when the piglets are born, right? They are born completely free of E. coli, but then they get contaminated hours after being born, typically from the dam, from the mom, or from the environment. E. coli is ubiquitous, present in the environment as well. So they get contaminated right there, and then obviously, depending on multiple factors, they're going to develop disease or not, right? But in this post-weaning phase, they also ubiquitous present in the environment. So that's where pretty much most of the time they get it, or either from their pain mates or from the dam that for some reasons could be excreting, shedding that at the weaning phase. And then they get contaminated or recontaminated at this phase, either from the new nursery that are getting in, or wean to finish, or even from the dam at the weaning phase for multiple reasons and all the stressors that inevitable happens right at that phase. And then yeah, they getting if they get infected, contaminated and get infected with these more pathogenic ones, so they have you know higher chances of developing the disease.
Okay, so now if I'm a producer and I find post-weaning diarrhea like in my pigs, like how would I be able to figure out if this is colibacillosis or if it's something else?
Typically, to diagnose colibacillosis, what we recommend is to either use antemortem samples like fecal swabs, and then to be more complete and really be able to assure that it's E. coli, we want to see intestinal attachment of the bacteria, because if you go out there right now and you swab pigs, you're going to find pathogenic and non-pathogenic E. coli in all of them pretty much. Obviously, if they have clinical disease, they're going to be shedding more of the pathogenic ones, but it's not uncommon to go there and kind of find pathogenic ones circulating, but the herd is clinically okay and good and thriving, So for that, based on that, we tend to associate at least 2 lines of evidences to confirm colibacillosis, which typically include histological evaluation of jejunum and ileum. So we want to see bacterial attachment at the superficial of the villi of the small intestine, plus bacterial culture, where you can, culture and have a single colony, and then you can use that colony either to do antimicrobial susceptibility and also to do bacterial genotyping, which is a molecular test that we use to identify those fimbriae and toxins genes that we discussed. So that become like actually 3 lines of evidence at the end, but I think with that we can really confirm E. coli. And without colonization, without seeing histologically the colonization, we are not as confident to tell that. But at the end of the day, clinical disease that comes to us as scour, diarrhea, right, at the phase, and then we do histological evaluation, we see colonization, and then we culture and isolate the colony, and then we send that colony out for genotyping, which is this gel-based PCR that we use to identify those toxins. So with that, we are pretty confident. Okay, we have a isolate that contain the toxins, fimbriae toxins, plus colonization of the intestine, plus the clinical disease in the field. So yeah, we are pretty confident that these animals are acutely infected, and E. coli is the cause. And that it's not uncommon to have some viruses underlying that bacteria infection. So we very often, test them for rotavirus, sapovirus, and coronaviruses as well. Obviously, we also rule out salmonella and other potential bacteria. But yeah.
And then, so once you get the diagnosis colibacillosis, like what can we do about it?
Yeah, that's a pretty good question, and typically it's depending of the severity of your cases and all that, you want to have your nutritionist in the table with you, with the report saying that it's colibacillosis. Okay, now we want to talk to the nutritionist, we want to talk to the site manager, and we want to better understand the other factors that could be associated with that, right? Like, was there any changes recently in the diet of those piglets? Was there any change with the barn staff that was taking care of those pigs? Any kind of factor that could be related to changes in either diet or management or transportation, how they were, how they took care of the transportation of those animals, how is the water quality of that farm, and how is the feed has been provided to those pigs. I think it's pretty complex and multifactorial, but you want to make sure that you have all of those things align in a better way you should do the best that you can do to provide to these pigs a cleanest environment as possible, and with water with high quality to them, and then feed as well. I'm not a nutritionist, but we have some nutritional factors that could contribute to the development of the disease as well. And so I think all of that should be considered if you have a case of severe colibacillosis or moderate or severe colibacillosis. And then from the pathogen standpoint, you can try to improve your biosecurity for those farms that you are facing the outbreak, right? And it's pretty hard to eliminate, but if you work on those factors that contribute to the disease, you're going to be able to control it. Like you still have probably the pathogenic strain circulating, but if you work on the other side of the factors, in the other factors that you have, you're probably going to be able to control the disease and control, and then you start working after that in prevention, right? Preventing the animals to get sick with that, working in all those factors constantly, I guess.
So it's not just about eliminating or trying to eliminate the bacteria.
Yeah,
it's more like getting all the other factors in place so that they just, even if they get in contact with it, they don't get sick from it.
Correct. And we have vaccines out there that are possible to obviously utilize as well, right? So you can kind of vaccinate and generate immunity and work in those factors in combination with that. And then, you can, from the biosecurity standpoint, and to prevent new outbreaks, you can obviously, let's say that you have a couple of sites where that more virulent strain circulating, you could increase biosecurity measurements and also practices on those farms. You could do not send those pigs out to any other sites where you would be able to spread that out even more, right? You could kind of direct those pigs to specific win to finish sites where you could do a better job preventing that to spread out and also controlling that. So you could kind of do those kind of things to prevent spreading that out in your system, and also to control the disease, I would say.
So is there anything else that you would like to talk about when it comes to the research that you've been doing on colibacillosis?
Yeah, I think in summary, I would say, we have enough or very good tools right now that we can better understand the pathogen and all the virulence factors and all that they are carrying along. And that helps us to obviously work in the direction of controlling the disease, and I don't think we're going to be able to stop that, obviously, but knowing your enemy is the only way to kind of fight him, right? So we need to keep doing that. I think we need to more and more fully characterize E. coli isolates that are associated with severe or more severe clinical diarrhea. And then from there, keep, having a better understanding how they are, becoming more and more virulent. And then perhaps in the future, we're going to have ways to either, not stop, but at least reduce that or work on that to avoid, spreading that out, or even having severe cases and outbreaks out there. We are focusing more on the pathogen at this point, but we have so many other opportunities when it comes to colibacillosis. Like I said, we have some nutritional factors that could contribute to that, and we have a lot of data about it out there. And also, obviously, we know modern farms, they are bigger. We have a higher number of animals being confined and all that. So sometimes the size of the pen is different in different farms and all that. So a lot of factors that I think we're still learning, and that could contribute to the disease. And I think we have a lot to learn still, and to understand from that standpoint, and that's going to help us to in the future, prevent disease, I guess.
So will you continue to work on this topic in your postdoc after your PhD?
That's the plan. I think like I said, we have a lot to do. I want to keep characterizing E. coli from now on, and then over the years we're going to more and more understand factors that contribute to this acquirement of genes and all that, and once you understand better how that happened, you can start to work to prevent it to happen, right? I believe so. Yeah, that's the plan.
Perfect.
It's time for our famous three.
Still delegating work to someone who hasn't adapted to AI? You don't need another assistant stuck in 2015. You need a remote AI specialist who can handle inboxes, research, reporting, ops, admin, sales support, and marketing, using tools like ChatGPT, Claude, OpenClaw, and automations. Wise VAs connects you with AI-trained Latin talent in your time zone at up to 80% less than a US AI hire. Schedule your free strategy call at www.wisevas.io.
Okay, well, to our listeners, if you're enjoying this episode of the Swine Canada podcast show, please remember to like, subscribe, and share it with your colleagues, friends, and family. So that then gets us to the final three questions. So the first question would be, what is your favorite swine-related book or resource?
Yeah, that's a good question, I guess. Probably you've heard of that answer a lot, but most of us that work with pigs, and especially with diseases, we have Diseases of Swine as a reference book, and I do. It's by far my favorite book. I'm always going back to that book and refreshing myself about different diseases in pigs, of course, not only E. coli, but I think it's a book that are heavily based on literature review, and from that book you also get a lot of good sources to go back in those papers and research in that research and go deeper in different topics in about different pathogens. So I think it's by far my favorite one because it's very scientific.
Yeah, like I do get that response a lot. So it sounds like it's a really good book.
Yeah.
So then what about a favorite book or resource outside of agriculture?
Yeah, I think, I have a couple of books that I read that I really liked. But one that, made a difference, I would say, in my life was Sapiens, A Brief History of Humankind by Yuval Noah Harari. He did a really good, research trying to understand us as humans, and like way back in time, many years ago, comparing us with even Neanderthals and all that. And I think he has a lot of like scientific explanations in those books that help me to even understand some behaviors that we have as humans nowadays, and that keep like passing through generations, and that kind of helped me a lot to understand, okay, that's why we tend to behave like this and that. So I kind of really recommend that book.
Okay, that sounds like a really interesting one. So I'll have to look it up. And then the last question is, in your opinion, what sets successful swine professionals apart from those that are not?
Yeah, that's a hard one. I feel like that one probably you have very different perspectives and very different answers. If you ask 10 people, you're gonna have 11 different answers in that one probably. But to me, I had great opportunity to have different mentors. When I graduated back in 2013, I worked with a food animal company down there in Brazil, which I learned a lot. And then after that, I worked with with a genetic company, with PIC in Brazil. And then here at Iowa State, also different mentors. I think one thing that I like about the ones that impacted me the most was the passion, being passionate to what you do. That kind of, it's contagious, right? You kind of, when you have a leader or a boss or a friend or a mentor that it's passionate with what they do. That's kind of contagious, and that kind of motivates you to keep doing more and more. And I think another really important thing to me is like communication. So clear communication of expectations. When you have that, you tend to disappoint them less, right? Or you tend to think more about what you're doing. So clear communication to me is a big differential for a successful leader. And we have other things that I think I want to keep working that myself, but I think being able to listen more and more before speaking, obviously, or genuinely listen to what people are telling you, right? It's a good skill to have, and an important skill to have as well. So listening more, and being able to ask the right questions. It's another skill that I consider really good, and people that can manage that, they are typically very successful. I worked in the field for, like I said, almost 8 years, and when in the beginning you go to a farm and then you ask so many questions and you barely listen to the caretaker, right? And once you start to getting more experience, you start to learn to visualize problems more, and see them without even asking questions, and saving the questions to ask the right ones, and listen more. I think listening more and asking the right question is not easy to practice and do that, but for sure, to me, I think it's a good skill to have, and whoever managed that well, it's successful.
Yeah, I like that answer. Like active listening, I think it's called. And active listening it sounds a lot easier than it is. Like
Exactly.
we tend to throw our own thoughts and ideas in before so we stop listening to the other person before they're done talking. So yes, it's definitely a great skill to have. All right. So thank you so much for joining me on the Swine in Canada podcast show today.
Thank you for having me again. It was a pleasure. It's always a pleasure to discuss and kind of summarize the research we've been doing.
All right. So that concludes another episode of the Swine in Canada podcast show presented by Wisenetics. So don't forget to subscribe to the podcast to stay updated on future episodes. So thanks, everyone. And until next time.